Hello, my name is Tanner Dabbert. I'm an undergrad researcher working in Dr. Ravenscraft's microbial ecology lab. My presentation is on characterizing the bacterial gut microbiota of herbivorous insects. Insects and plants have co-evolved over millions of years, with herbivorous insects having evolved the ability to strongly resist and metabolize the plant defensive toxins. Previous research has linked insect-associated gut bacteria as possible participants in this detoxification process, and our primary research targets as potential toxin-degrading symbionts. In order to identify these toxin degraders, identifying the gut-associated bacteria we can successfully culture in aerobic conditions to be used in future research is of extreme importance, as we have defined methods for identifying toxins in aerobic cultures. Through the study of the herbivorous insect orders, grasshoppers, beetles, and caterpillars, we aim to characterize the gut-associated bacterial communities as well as identify what is able to be isolated in culture. These would be significant findings as a majority of the mammalian gut bacteria is not cultivable, and most that are require anaerobic culture methods. We expect the insect gut microbiota to be easier to culture, largely due to the majority being derived from the environment and housed in primarily aerobic insect guts, thus living in aerobic conditions. The methods used, as seen in the diagram here, included dissecting and homogenizing insect guts, and then either using 16S Illumina sequencing to provide a wide overview of the bacterial genera population contained in the gut, or plating and isolating bacteria to be identified through Sanger sequencing and NCBI BLAST programming that assigned bacterial identities to our isolates based on sequence similarities to previously sequenced and identified bacterial genera. This figure here is our Illumina data, with each line representing a single sample from a species in one of our target orders. Each color represents a bacterial genera that is identified in the legend on the side. Each sample line is colored correlating to the relative abundance of each genera found in the gut of that sample. We can see visually these composition trends of the overall gut bacteria community of each dissected insect and can identify the most prominent genera in our species samples, or when compared with samples from the same order, we can see trends at the order level. Here we can visually see the most prominent genera in grasshoppers being Weissella, Spiroplasma, and Enterobacter, Pantoa and Enterobacter in beetles, and Cutibacterium, Staphylococcus, and Pseudomonas in our caterpillars. The table here shows our data relating the identifications from the isolate library we created with the data from the Illumina sequencing. In the second column is the number of bacterial genera that were found in greater than 20% of insects in a given insect order in the Illumina sequencing results. These were classified as frequent bacterial genera. Then this third column shows the number of these frequent bacterial genera that have been identified through isolation. We can compare these two columns to see how many of these prominent genera we have been able to culture. The last column is the percentage of Illumina reads per insect order that matched one of the isolated frequent bacterial genera. This last column is a good metric showing how significant of a portion this table is concerning by illustrating just how much of the bacterial relative abundance has been sampled through bacterial isolation. The isolate data shows us that a significant amount of gut bacteria is likely culturable in aerobic conditions, and in conjunction the two give us an idea of the relative abundance in vivo of the isolates we have obtained. The bacterial library of in vitro culturable isolates that have been identified can be compared with the genera identified as prominent through the Illumina data to give us primary targets for possible toxin degraders, as these prominent genera having larger abundances likely indicates a functional importance. These can be tested in future research through the implementation of in vitro toxin degradation assays. Subsequent identified degraders then can be tested for in vivo detoxification and importance to overall insect metabolism. Alternative strategies can also be developed for potentially culturing more of the genera of bacteria we have recognized, but have been unable to isolate up to this point. We can look up culture methods specific to these genera, go out and dissect more insects, and try to grow those bacterial genera using the specialized methods. This project will provide insights into the role of microbiomes in herbivorous insects' ability to survive on diets of toxin-defended plants.